Bonjour, bienvenue. Thanks very much for joining us at cette conférence for chaque semaine. And we are absolutely delighted today, with someone who is going to be central in terms of ongoing theme on precision medicine. But before I introduce our esteemed speaker today, just a few logistics as per our other sessions. And please pose your questions in the Q&A section on the bottom of your screen. And you can do this as the lecture is progressing, and we'll answer them in turn at the end of the lecture. In addition, you could actually join directly in the Q&A session in person by posting your questions in person. And so you can raise your hand and we'll invite you into the panel for that opportunity. And if you do have any technical issues, use the chat box to indicate the issues. And then Allison or Kelsey will be able to touch base with you to make sure that you can join us successfully. And today we are absolutely delighted that Dr. Arvind Mer in the Department of Biochemistry, Microbiology, and Immunology is able to join us for a really exciting lecture. He's also a member of the Ottawa Institute of System Biology in the Faculty of Medicine. And Dr. Mer obtained his master's degree in computational and system biology from Jawaharlal Nehru University in India. And then he transferred to Germany after he was awarded the Helmholtz Graduate School, International PhD Fellowship at the very famous Max Delbruck Center for Molecular Medicine in Berlin. And so I know some of the folks there, and they were some of the earliest pioneers, certainly in transgenic models. And we have continued to collaborate with them. In 2014, Dr. Mer completed his PhD studies and moved to Sweden to undertake the postdoctoral fellowship in bioinformatics at the Karolinska Institute in Stockholm. And in 2016, he came to Canada and took up a senior postdoctoral fellowship, and then subsequently as an affiliate scientist at the Princess Margaret Cancer Center at the University of Toronto. And he was recruited to the University of Ottawa in 2021 to the Institute of System Biology and continue his current endeavors in bioinformatics and in solving major biological problems. Dr. Mer's lab is using cutting-edge machine learning and artificial intelligence technologies for omics data mining to advance personalized medicine. And he's utilizing these approaches to help to fast-track the insight and knowledge of molecular mechanisms of disease and biology. Dr. Mer is particularly interested in the personalized medicine and also using single cell sequencing data analytics, and also the ability to identify novel therapeutic targets, and possibly even using repurposed drug profiles. And so we are absolutely delighted today and look forward to his lecture entitled Machine Learning for Precision Medicine. Thank you so much, Dr. Mer, for joining us today.
Thank you for the nice introduction, Dr. Liu. I'll share my screen. I hope we could see it.
Yeah, it's perfect. Yeah, okay. Please go ahead.
Okay, thank you for again, thank you for the kind introduction. And my name is Arvind, and I'm going to talk about machine learning for precision medicine. So to begin with, first question is, what is machine learning? And we use it. I use it a lot in my research, but when it comes to defining, it can be sometimes really confusing because there are so many names going around, like artificial intelligence, machine learning, deep learning, and so forth. So machine learning is a broad term that we use for methods and algorithms that we use for the artificial intelligence techniques. And deep learning is a subset of machine learning algorithms. And artificial intelligence is a broader term that can be applicable from robotics to the medicine to any field where we use computational intelligence for the analysis and decision making. I'm going to use machine learning and AI interchangeably. Okay, so what is the intelligence or what is the AI? Intelligence is the capacity to learn and solve problems. We see some examples in the real world, and then we learn from them, and then we use that knowledge to solve the problem. And when a computer is doing this thing, this is called artificial intelligence. You have seen the applications or the buzz about the AI in different fields. It's being used from social media to finance to online shopping in all these different places. And of course, there is a lot of talk about using AI and its applications in the healthcare also. Now, in practice, how does it exactly work? So you all might have seen these kind of screens where you see one particular show or one particular movie, and then Netflix or Amazon or other streaming services, they recommend you similar movies or similar shows. And this is all done by AI in the background. So to give you a more practical example or a simple example, let's say a friend comes to you and says that I have seen these 3 shows, and that friend is asking you for a recommendation. What should I watch next? So you have these 4 options or 4 choices here. I mean, in your mind, in your head, just imagine what will you recommend based on the knowledge that your friend likes these 3 shows. So last time when I did this kind of analysis or this time kind of quiz, uh, a large number of students replied using an online system. I don't have it right here, but I guess we all kind of make a guess what the right answer might be. So, but when you're present with this knowledge, or when your friend comes with this information that I have seen these 3 shows and I want a next recommendation, you first learn something from these shows about the choices, about the interest of your friend. For example, you can simply see that all these 3 shows are animated shows. So the user likes the animated shows, and these are comedies. So you can say, okay, the user likes comedy. They are not kids oriented or oriented for children. So you can say, okay, the user is an adult because he's liking a show which is more geared towards the adults. And this is what any machine learning algorithm is doing in the computer. So basically, computer is learning these rules or deducing these rules from the presented data and building a model. And this is what we call building the model. Now, based on your model or these rules, you can score each of the shows, the options that you have. For example, you can say the user like animated shows, but this particular show is not an animation. So you can give a -1 point, for example. User likes comedy, so you can, this is not a comedy, it's a reality show. So you can give it a -1. It's not for the kids, so you can give it a +1, and user is an adult, so +1, and so forth. And similarly, for some other show, you can rank the show, or sorry, you can give it a score to other shows also, where what the show is geared based on your model. And at the end, you can have a score for each of the show. And based on these scores, you can decide, okay, this is my highest performing show, so this is the what my recommendation is, and hopefully that's what your recommendation is. So to summarize, this is what the machine learning is, and this is what's happening behind the scenes in the computers. Whenever we talk about AI machine learning, basically the computer is trying to learn from the patterns in the data, and based on those learned models, it's trying to uh, to recommend or trying to decide for a new data set. Now, second part of my talk is this: what is precision medicine? So I just have only one slide which summarizes the precision medicine, and I hope it summarizes well. So traditionally, the approach have been in medicine is that if you have a patient from a disease, let's say breast cancer, we treat them in the same way, or it was thought that all the breast cancer patients are have a same kind of disease. But in recent decades, the genomic has made it clear that in cancer, for example, in breast cancer, not all patients have the same kind of cancer, or their genomic profile of the patients is completely different. And then comes the question, can we decide the medicine based on the patient's genomic profiling? So basically, you can have genomic profile, different kind of omics profile from the patient, and then you can analyze the data, and based on that, you can decide whether a patient is a responder to a drug or not going to be a respond to a drug. And you give the drug only to the patients that you know are going to respond. This is basically this is in medicine. Now, what are the challenges, or what on paper this idea sounds great, but there are a lot of challenges. For example, this is the data from several big clinical precision medicine related clinical trials in cancer specifically. So here they try to match patients to a drug or a clinical trial based on mutation profile only. And as you can see, all these red bars here represents the percentage of patients that we could not match to any clinical trial or any drug basically. So as you can see, these bars are quite high. So one of the big challenges in precision medicine is to find these biomarkers which can be match a patient to a drug based on the genomic profile itself. And in that context, my research is based on. So what we do is that we take the different kind of omics data as well as we take the drug response data. So most of the time these are cell lines, for example, or patient-derived xenograft models that I'm going to talk soon more. All these different kind of models, or the cohort-based studies. And then we develop the tools and techniques and databases that can format this data and can make this data useful for the analysis. And then we apply machine learning on those data to find these responders versus non-responders, or finding the drugs which can be more effective for a certain kind of patient population. So in my talk, I'm going to give one example from each of these groups. So first, talking about tools for the data analysis. So specifically in cancer, so most of my work has been focused on the cancer. So I'm going to talk about the cancer examples. So in cancer, we have different kind of model systems. There are cell lines, for example, cancer cell lines, which have been the workhorse of the research and used frequently. There are organoids, cancer organoids that are three-dimensional model of the tumor. Then there are patient-derived xenografts, they are in vivo model, and then you can also use the clinical or patient data directly. However, there are limitations of each of the model. And we have realized in recent years, we have realized that the cancer cell lines are not the best model that represents the patient. And PDXs or patient-derived xenografts and organoids are known to better recapitulate the patient's biology. So there's more and more focus to use these PDX models into the research. So for example, a recent recommendation from Cancer Moonshot project is also to develop these 3D models. So what happens in this patient-derived xenograft models? So we take a patient sample and we implant it in a tumor sample and implant it in an immune deficient mice. Because this mice is immune deficient, the tumor grows there. And at one point when the tumor volume is large enough, you can basically sacrifice the mice and chop the tumor into pieces and implant it into new mice. So this way you have more and more tumor material. And at one point when you want to test a drug, for example, just like a clinical trial, let's say you have a 6 PDXs, you declare 3 as a control and the rest 3 gets your drug. And you measure the tumor volume every week or according to the protocol. In the best case, we are hoping something like this, where in the control group, the tumor volume is increasing, while in the treatment group, the tumor volume is decreasing with time. And in PDX pharmacogenomics, our aim is to connect the genomic data from the PDXs from the tumor with the drug response data here and discover the biomarkers which can indicate whether a drug is, what are the biomarkers for this particular drug, or whether a patient is going to respond to a drug or not. So for the analysis and management of this particular data, patient-derived xenograft data, we developed a package called Jiva that connects the genomic data with the drug response data. This work had been published in Cancer Research. The package is freely available on GitHub as well as in Bioconductor R repository. So in short, what we do in this package is that we take different kind of genomic data, we standardize all the data because there are different methods of analyzing genomic data. So we use the standardized methods so that across data sets we have uniformity and so forth. And then we connect this data with the drug response data. Once you have this Jiva set or this particular object, you can do different kind of analysis, starting from plotting the data, visualizing the data, computing different kind of response metrics to biomarker discovery and machine learning also. We also have functions in the package to directly pull the data from cloud storage also, and using from different tools that are used in the lab. So just to give you an example, this is an example of a real world data, PDX data that we have. Just using a small line of command in R, you can plot this kind of visualization. Here, each dotted line represents one individual PDX, a solid line represents their average, and the red line are for the control and the blue line is for the treatment. And you can change this visualization, you can normalize them, and you can compute the error bars, etc. So it helps a lot when you are doing large scale studies and trying to manage all these PDXs. It can quickly compute all these different matrices and different visualizations. Another big challenge in PDX field was still is that a response metric. So people who have worked with the cell lines and the drug response might know about IC50 or area under the curve is commonly used for the cell lines. However, for the PDXs, there is no such unified metric. So we have implemented several different matrices in our packages. You can compute angle between the treatment and control curve. We also have functions to linear mix model and so forth. Okay, to give you an overview of how these what the package provides for the large scale clinical PDX-based clinical trials. Here is an example for the breast cancer PDXE dataset that we show. So you can generate these kind of plots with simple commands. Now, what you see in this plot is that each box here represents the drug response in the PDXs. In terms of response, if it's red, it means there's no response. If it's blue, it means there is a response, we see a response, basically. These are different drugs that have been tested. Each column represents a patient or PDX from a particular patient, and the first row is the untreated or the control. And as you can see in the uncontrolled, we expect it to grow fast or there is no response. So you see mostly the red. And then you see there are some of the places where we see the responses, but mostly there are no responses. So these kind of plots help us to find out such a scenario where a particular patient is not responding to almost any of the drug, while some of the other patient is responding to a lot of different drugs. So when you are doing a large scale PDX-based clinical trial, these kind of plots give you an overview of the experiments and how it's going, and how much response you are seeing in the data set. We also performed biomarker discovery because we have the genomic data, we have the drug response. What you see here in this side is the gene expression profile for selected genes in the PDXs. And on the right side, you see the concordance index between the gene expression and the drug response. Each dot represents the effect size, basically, or association between a gene expression and the drug response, if it's statistically significant. So to highlight an example, we have found that ERBB2 gene expression is significantly associated with trastuzumab drug response in breast cancer. Now, and we also looked at the pathway association between drugs and the genomic and the pathways in the cell. So for this analysis, we classified the drug based on their target into different groups. For example, all the drugs that target MAPK gene are grouped here together. And then we do the gene set enrichment analysis and different pathway analysis. And again, to highlight some examples, we have found that MAPK class of drugs in PDXs are targeting MAPK activation pathways and sorry about that, MAPK class of activation pathways. So this gives us an indication that in PDXs you can target the correct pathways using these different drugs. Now, recently using this data set, we have trained a deep neural network to assess the growth rate of a tumor. So basically we take the gene expression data from the PDXs and the tumor growth values from different PDX tumors, and we train the network. And then when we apply this particular model into different real world TCGA, for example, data, we see a statistically significant difference in many cancers between the high growing and low growth rate tumors. So now coming to the machine learning for patient to drug matching problem, the second part of my talk. Here we would like to give the example of acute myeloid leukemia. So it's a hematological cancer. The 5-year survival rate is about 25%, which is quite low. And it's defined by a long list of mutations that can be present in the patient. Among these mutations, mutation in the NPM1 gene is the second most common mutation. It's an important mutation that it's considered a separate class. However, there are indications that this is a heterogeneous group. So we decided to look into this particular group of patients. And at UHN, we had a cohort of 77 patients for whom the NPM1 mutation was present in all of them. And we perform RNA-seq on those patients. And then I did the machine learning analysis, clustering analysis, and found that there are two groups within this patient cohort. So to see whether these findings hold in other data set also, we took the TCGA data set, which is completely independent from us. And there also we applied same method and we saw the same kind of pattern. We even went further and I got the data set from Karolinska Institute, and in that data set also we see the same pattern. We also established collaboration with the folks at McGill, and in their data set also we see the same pattern. And another data set called BTML, we see the same pattern. So basically in 5 different data sets, we are seeing same pattern. And then we assessed what are the differences between these two clusters, because now we know we have 2 clusters, but we want to know more about the properties of these 2 clusters. So when we do the cellular deconvolution analysis based on the stem cell score, we find that in one of the subtype or in one of the group, we find that the stem cell score was quite high, indicating that it's in the primitive state of the maturation. We call this one primitive cluster and the other cluster is called committed. Now we perform the differential gene expression analysis across the data in different data sets individually, and then we combine them into using a meta-analysis approach into one value basically. And what we see is that there are certain genes. For example, we found that HIP and CDH2 and GPR12 are highly expressed in primitive cluster. On the other hand, genes such as CDK163 or C1qa are highly expressed in the committed subtype, and they are related to immune process. We also looked at the chromatin accessibility landscape of these samples, these subtypes. Here we performed the ataxic analysis on these samples, and then we did the clustering. So this is the ataxic based clustering, which perfectly may match with our gene expression based clustering. So it shows that there is a very good synergy between the ataxic profile as well as the gene expression and gene expression profile. And we have shown that the primitive and subtypes are enriched in promoters, while committed subtypes are enriched in the intergenic regions. Okay, so we also looked at the survival difference between primitive and committed subtypes. The primitive subtypes have a poor survival, as you can see here, and this is also significant in a Cox regression model. Now, we have established that there are these 2 subtypes within NPM1 group, we ask the question, can we find the drugs which can be helpful for these primitive subtype patients because their survival is worse than committed? So to find the drugs which can improve the survival in the primitive subtype, I developed a machine learning pipeline. So this is a little complex example. There are a lot of things going on, but basically, in short, what we did is that we took the cell line based pharmacogenomic data, which is a big data set, CCLE data set. And based on that, we trained a machine learning model to predict primitive and committed subtype. And then we ran our UHN data set through this pipeline and predicted the list of drugs that can be effective in the primitive subtype. And then we took some of the, so here is the list of the drugs, and these are the drugs that were on the top of the list. And then we took some of the top drugs and tested them in the lab in ex vivo setting to see whether they really show a difference or not. So this is the drug dose response curve, for example. As you can see, these are more effective on the primitive compared to the committed in our data set. And we also used an external BTML data set to verify that this is not our data specific results. Rather, we see this pattern across the data. And we also tested, we tested basically 5 drugs, and we found that 3 of them showed this pattern in both the data sets. So it was quite robust signal. Sorry about this. So this work had been published in Nature Communication recently, and based on these, we are designing a basket clinical trial where we are trying to recruit the NPM1 mutated patients, and we are advising to include these kinase inhibitors into the drug regimen, into the treatment regimen of these particular NPM1 mutated primitive subtype patients. So I'll just take few more slides to talk about my future research. So in my lab, we are working on basically 3 main aims we have. First is that tool for precision medicine. We will continue to develop these R softwares in R and other programming languages, which we make available in an open source environment to community to use. These includes different tools for the genomic data analysis, single cell data analysis, and so forth. We are also focusing on drug repurposing, where we are going to utilize the approved drugs for different diseases. We are also focused on multi-omics data analysis, that how can we analyze and extract more information from different kind of omics data sets. Talking about drug repurposing, you might already know the fact that drug development is very expensive and time consuming. It is approximately takes around 12 years and $2.6 billion to develop a new drug nowadays. There's a lot of discussion about whether this model is even sustainable for the long term for the drug discovery. So anyway, on the other hand, we have this this approach of drug repurposing, which try to identify new use for the approved drugs, or which are investigational drugs that are outside the scope of original clinical indication of the drug. There are a lot of advantages of this approach. For example, you don't have to go through all those clinical trial phases, and because you have a lot of information already available about these drugs. So to give you example of such a pipeline where we are thinking about is, okay, for example, you can take a healthy and diseased tissue, and you can do a data-driven comparison between them to find out what are the genes, for example, or regulatory functional modules that are dysregulated between the healthy and disease. And then using the pharmacogenomic data, large-scale pharmacogenomic data, or protein-protein interaction network, or human cancer cell atlas kind of projects, you can build machine learning models which can tell you that what are the drugs that can revert the gene expression pattern, for example, from disease to healthy. So this can give you a list of drugs or prioritize certain drugs that can be further tested into the experimental setting in the lab, for example. Now, another, as I mentioned, one of our focus is also integrating multi-omics data types. So when we talk about omics, usually people understand the classical omics like RNA-seq, CNV, or mutation, methylation patterns, etc. But there is large amount of data available in terms of radio images or electronic medical records, and we are trying to integrate these different data sets and trying to understand what is the relationship and association between these different data sets from the machine learning point of view, and how can we utilize these data sets also for the precision medicine. So to summarize, selecting right therapy for each patient is crucial for precision medicine. Biomarkers predictive of drug response are needed to increase response rate and decrease toxicity. Analysis of high throughput pharmacogenomic data is challenging. Uh, AI and data science can offer an opportunity to advance precision medicine, and ultimate goal is to develop biomarkers that can better translate to the patient. So at the end, I would like to acknowledge all the people who have helped me throughout my journey, and thank you very much. And if you have any questions, you're welcome. And you can send me email also and my email address. Thank you very much.
Yeah, thank you so much, Arvind. This is a fantastic, very, very timely, as I have mentioned earlier, so I should mention that Dr. Mer and I had a chat before the lecture, and, very, very exciting. And, we know a number of mutual friends, but at the same time, I know Dr. Mer is going to be talking with some of our investigators as well. So this is super exciting and very timely. Because of our precision medicine priority initiative, and currently, we're actually putting out the pilot funding opportunities for people to collaborate. But this is a really ongoing process. So we want to actually continue to build on the accomplishments of people so far. But so we have a great ambition, and so I think Dr. Mer's expertise comes at the perfect time. And so maybe we can have questions. I certainly have a number of questions, and this is an exciting, evolving field. And one question is from Dr. Rob Beanlands. And so he is, he's a chief of cardiology, but also he is an imaging expert as well. And so, he really enjoyed the lecture, but he was wondering in terms of what are the, I guess, pros and cons, of the different, I guess, AI tools, that's available. So certainly their group has done a lot of work on the image analysis, I guess it's radiomics, and, so they use a variety of tools. And so the question is, when you are looking at the different machine learning or deep learning protocols, and what are the advantages of these fancier tools? Do they really provide sort of a real value, or is just I guess AI gymnastics? What is your view on the tools available?
Actually, this is a very interesting and important question in the field in general also. So it has happened many times with me when I was in Toronto, that some of the, for example, students coming from the university, bachelor students, and they are saying that, I have heard about this fantastic AI method. Can I apply it, or can I basically cure the cancer using this method? And I always tell them, okay, you can do it, but let's start from the regression, or let's start from the simple method and check whether it works or not. So my principle is always start from something simple and then go to the complex model, because it's easier to say that, okay, my complex model gives me good performance, high accuracy, and so forth. However, what are the trade-offs we have to be always aware of? With the complex model, they are difficult to explain. They become a black box model. And in our field or in medicine, basically, this black box approach is not much appreciated, and we don't want that. Basically, it's a one thing if you have a black box model for movie recommendation. Nobody cares why this model recommended X movie or Y movie, because it's okay, it can happen sometimes. But in medicine, it's a life and death situation. It's a completely different aspect. So with the increasing complexity comes this black box thing. So we have to be aware of this. And my approach is always start from something simple, which is simpler to explain, and then move forward to the complex model.
Yeah, fully agree, because sometimes, the fancy models can overfit and pick up, some funny artifact, data pattern that has no, relevance to the real world.
Yeah,
it's a fantastic fit. You can show off, the area under the curve, but really has no practical meaning, at the end of the day. Yeah. Yeah. So I really like the approach that you have. Yeah. So be sure you post your questions, in the Q&A, or if you want to actually ask the question directly, let us know. So the question I have is that we do a number of those projects that you recommend, sort of comparing the healthy and the disease, sort of different types of controls, and as we come up with a lot of potential candidates, right? So the big question always, and this is where a lot of the effort of our scientists is which one's real, which one actually matters at the end. And so until you do that clinical trial, you really don't know, right? So
yeah.
So the challenge is whenever you do the reductionist models, right? Whether I think you mentioned, whether you do cells or organoids or animal models and things like that, there are always trade-offs and the things like that. And so what we have found sometimes is that these databases may actually give you contradictory information, and nobody really knows what's right, what's not right, until you do, of course, the clinical trial. But then everybody's trying to avoid that, or try to be smarter going in rather than going blind. So how have you kind of approached that, because certainly you have done nice grids, try to do the analytics, and but for breast cancer, trastuzumab is not kind of the biggest thought on there, right? There are many other candidates which probably don't pan out, right?
Yeah.
And so how do you kind of sort this through to know what's what's real, and how do you differentiate all these contradictions sometimes in all these data sets?
Yeah, I mean, this is a big thing that we deal in day-to-day research. These false positives, that what we are seeing, are they really the truth that that is there? And the ultimate test is, of course, always the clinical studies or clinical validation or validation in the lab. However, we can increase the amount of evidence, let's say, for a lack of a better word, using different other computational approaches, like looking into the pathways that, okay, as I showed in one slide, whether these drugs are also targeting the correct pathways or not. So we can look into that, for example. Similarly, you can look into the association between, if you have a biomarker in context of genomics, if you have a biomarker, what are the relationship between this particular drug and the biomarker, whether they are doing molecular docking kind of studies, and looking into whether they are actually related or they are interacting with each other or not. So there are multiple computational bioinformatic based approaches that we apply to make it more evidence is more stronger, or as much as we can make it stronger. But of course, there is a big challenge that what we do in one data set, sometimes we don't see the same pattern in other data sets. And there are multiple reasons for that, starting from the technology, what kind of technology you are implying, what kind of technology is applied to generate the data. Microarray data is completely different, can have different patterns than the RNA-seq-based data. So there is these kind of differences, and we have statistical method up to a point to control for these different variables, or we try to control for these variables through different statistical approaches, and that helps us to increase the amount of evidences.
Okay, yeah, so very, very thoughtful, yeah, because this is a conundrum we face, and certainly we think, not by any sort of scientific backing, is that it's really the ability to combine really very robust, analytic techniques and good methodology in terms of producing those data, but also the ability to use as much biological insights as possible, so that there is multiple ways of validation, just to make sure that the observed association, for example, is real rather than artifactual. Yeah. And so let me just see if any other question. Yeah. So in pursuit of digital twin. Okay. Okay. So this is an interesting question. This is from Dr. Rebecca Thornhill. And she does a lot of the radiomics work as well. And she really thought your talk was fantastic. And so the question that she had is interesting, is on machine learning and precision medicine. There is a kind of the concept of developing a digital twin for the patient. And is this at all a useful approach, for drug discovery?
Yeah.
Interesting question.
Yeah. Thank you for appreciating the talk first, Rebecca. And I'd say it is an interesting idea, but from a machine learning. Okay, so from my personal experience, what I love to have in any project is a vast number of samples, or looking through vast number of patients rather than having a lot of information about very small number of patients. So there is always this kind of trade-off, let's say, that if we have always have limited resources. So in context of cancer, if we have a grant money to sequence patients, we can either sequence at the single cell level 10 patients, let's say, or using the same money we can sequence 500 patients. And usually we go for the 500 route because this gives us also an idea about variation within the population, and how much the pattern is varying across the patients. And once you have this kind of data, then you can focus on the selected number of samples or selected number of patients and then go deeper into that. Same, in my opinion, personal opinion, same applied for the digital twin concept. You can have as much, a lot of data about one particular patient, but this comes with the fact that the cost is high and the system is dynamic. A patient is at one point, if you're sequencing a particular patient, again giving example from the cancer, the tumor doesn't stay the same in 1 month or in 1 year, because the drug is put in the pressure, and then you have to sequence again and again and again. And there are these kind of limitations that in my view, but definitely this is a great idea.
Yeah, so if we had the no limited in resources, we'll get digital twin on everybody. Yes, and so that's interesting, because you're absolutely right. Because quite often the tension is, of course, you have limited amount of funds available. And, so for example, for our institute, we wonder whether we should go with the approach of trying to get at least some sample on every patient that we see, versus have very detailed study on, let's say, a very, very select group. So I guess that the best approach, in fact, is kind of a combination of the two, from your point of view, so that we truly need to go broad, even though that might be shallow, and then, go deep on some of the key disease types.
Yeah.
Yeah. Okay. Okay. All right. So that's a very important advice. Yeah. And let me just see if there are other questions coming up. So I have another question as well from, I guess from my own point of view, and that is that um, here is, I guess, the conundrum that I face, and I wonder whether there is a solution to this, and that is that the deep learning, or any of the machine learning, are very good in terms of pattern recognition, right? From existing data sets, and being able to then apply that to the new one. Like your Netflix is a good example. But the information or knowledge continue to advance. And so use your Netflix model, so let's say, I'm interested, in picking out my next, sort of favorite movie, and, look for one that has like a 98% matched for my interest. But, I read an interesting article in New York Times today, on, let's say, I don't know, like the new Macbeth, and it really actually has and it's completely out of my range, and I really actually thought that might be interesting. So the question is, how do you incorporate, but then same thing, like you could actually have your beautiful pattern candidates, and then boom, another paper in Nature or Science, comes, it's relate to your area, but it's really actually a whole kind of a new twist on everything, right? It's not in your original database, but that's something new. So is there a way to analyze your data, sort of using existing pattern, of course, because that's what AI is really, really good at. So how does one actually incorporate, into the learning or model of something that is quite new, or, or maybe there's a, change in the in the environment or something like that, so and you need to take that into account. Is there a way to do that, so that you're not totally reliant only on past data patterns?
So traditionally, we rely mostly on the past data. But if I am Netflix CEO, I will say that give me access to your New York Times subscription so I can see what you are reading, and then I can include that information also into my model.
Okay.
But in terms of in biology, or in other practical applications, yeah, of course, there are methods called online learning.
Yes.
So where we try to evolve the model or retrain quickly the model based on the new information or new knowledge.
Right.
Yeah, and this is being used in the industry a lot. These online modeling, online learning systems that evolve with time or with changing patterns. But in medicine, it's still very limited, or in biology, the use is still very limited, because our data sets are comparatively small compared to all these Netflix and Amazon platforms, and it doesn't take too much to train the whole model using the existing data. And we are always a lot rightfully concerned about the variation in the data. Like if you have a model trained on patient population within the Ontario, for example, and then you get certain new samples from a remote region of the Ontario, it can happen that the population, genomics of population can be quite different. The ethnicity or the mix of the population can be quite different. So there might be a totally different variation there. And using our existing model for that kind of population might not be the best approach. So we try to always compare first whether the new data set is similar to my old data set. If there are differences, what are the differences? And can we take into account those differences also?
Yeah, so I think, sort of making sure that the conclusions are robust, and, so that you're not being misled as well. Yeah. And so the other question I have is that what is is there a parameter, so in precision medicine, so what we like to do is to define kind of a group of I guess a subpopulation, right, that share characteristics with each other, and then we will be able to potentially find patterns such that the treatment, be more effective, for this group. And so the question is, given that there's a high individual variability, sort of patients or people are messy, right? And so yeah, you don't have very clean, sort of sort of phenotype, so to speak. Even though we do have disease classification, but for example, the example I give, like heart failure, like aortic diseases. In fact, they're probably not single disease, right? They're all sub diseases, very similar to cancer. So, is there a reasonable I guess a size, of a subpopulation that, because ultimately we talk about precision medicine at the individual level, but that's not true, because you can't actually find a drug really, effectively using any statistical method to say that you actually got something. And so is there a kind of a size of a subpopulation, that you can actually have, let's say, quite thorough omics data, these type of things, and that when you, let's say, do find potential therapeutic candidates that will be large enough to say, this is real, and this is, the population that has these shared characteristics are likely, going to be responders, and we can then, launch a study on that. Because I think if the sample is too small, while it'll be very robust, but it's probably not going to be reproducible, the moment you take to the real world. If it's too large, then you kind of miss the point, right? Is there, from your experience looking at the cancer, where I'm trying to figure out what the most optimal, sort of subgroup size is, to really have that robustness, yet enough similarity that we can say, okay, so this is a reasonable precision group that we can actually go forward with. Yeah.
That's a really interesting question. So one thing I can remember is that in 2017, I published a paper where we looked at the effect of the sample size and reading depth, etc., in breast cancer subtyping. And there we showed that after a point, it kind of flattens. The performance basically of the model flattens in machine learning. So adding more samples after that particular point doesn't make much difference in there. I don't remember exactly the number, but I think it's around 100 or something for breast cancer specific problem. But yeah, as you said, it depends upon a lot of other factors also, like how much is the variance between in the population is present, how diverse or what is the background of the population is, and what is the nature of the disease. For example, if it's a just simple monogenic or depending upon one gene kind of disease, then it's easier to you can go with the smaller number of sample size. But if it's a complex phenotype that is arising from multiple genes or having effect from multiple mutations, for example, then you might need a larger population size. And that's one of the reasons that I always tell my collaborators that before you design the experiment, at least consult with us or talk to us, that before you are sending your samples to sequencing or anything, just at least let us know that how many samples you are sequencing. And so because there are situations sometimes people just sequence 2 or 3 samples and then they want to extract some information or want to do some analysis on that, which is quite difficult. Or basically, it's, as you said, it's not statistically robust, might not be reproduced in other data sets. On the other hand, spending money on doing large number of samples and then expecting it to uncover almost everything is also not feasible or not fair.
Okay, yeah, so that's actually good. So it's reassuring, a very, sort of wise insight, because you're right, for us, we collect a patient cohort with the samples, we're sure that, the next, the $1 billion drug targets is in here, please find it for us. But then you talk with people like Jeff Hinton. Jeff Hinton says that, no sample is big enough. If you have 100,000, well, give me another 100,000, like.
Yeah, I mean, from the deep learning point of view, So I said a number around 100 or something, but
Yes.
a deep learning person will be highly offended by that number. They are like, what are you talking about? The 100
Yes.
is nothing for them because, yeah. So it also depends upon the complexity of the algorithm. Going back
Yes.
to our question previously,
Yeah.
the previous question, that
Yeah.
what kind of model you are using and how complex the model is. So
Yeah.
that's also an important factor deciding what should be the correct sample size.
Yeah. Okay. So that's good. Yeah. So I'm very happy and relieved to learn of your study results and recommendations. Yes. So there's hope after all. Okay. So yeah. So maybe we can sort of take this opportunity to thank Dr. Arvind Mer for really a very very refreshing talk, up to date, and very relevant as well. And I think this is really the beginning of great collaboration. And we talked already with Dr. Mer in terms of potential interest groups, which we can assemble and then can work with Dr. Mer, because we want to make sure that we do have a small critical mass, for the group, so that there is adequate sample patient population, and at the same time, really questions, that are amenable, to answering with some of these state-of-the-art tools, and then, being able to follow up once the results are available, so that we can really, get to the impact that we're looking for together. And so this is fantastic, and I know you will get a chance to meet some of our groups. Hopefully that's just the beginning, in terms of the collaboration. And so we certainly, very much thank you for the wonderful lecture, and welcome you to the community. And look forward to lots of discussion going forward, and getting your help to sort out our data and the clinical conundrum. And there's no shortage of it.
Thank you very much.
Yeah, so take good care. Yeah, all the best. Bye-bye.
Thank you. Bye.